Hello everybody, this is the biology review for grade 10, semester 1 at the International School of Innovation.
This chapter is talking about the nervous system, nervous communication. It's about the organization of the nervous system in the vertebrates. What is the function of the nervous system? Mainly the function of the nervous system is to receive information, to process information, and to respond to this information. The central nervous system is composed mainly of two parts, the central and the peripheral nervous system. Here we have spinal cord, brain, and we have the nerves. Central general nervous system of a vertebrate. It includes mainly cerebrum, cerebellum, medulla oblongata, and we have the spinal cord. Cerebrum, cerebellum, and the medulla oblongata, they represent something known by the brain. Here we have a structure in brain or the encephalon, the frontal lobe, the temporal lobe. Cerebrum is composed of two hemispheres, the right cerebrum and the left cerebrum. We have the cerebellum here, and here we have the medulla oblongata. Finally, we reach to find the spinal cord. The nervous system is composed mainly of two parts, as we said, the central nervous system and the peripheral nervous system. Mainly the central nervous system is composed of the brain and the spinal cord, while the peripheral is composed of the cranial nerves and the spinal nerves. Cranial nerve, they come from the cranium. Brain is composed of cerebrum, cerebellum, and medulla oblongata, as we said before. And the cranial nerves, they ramify from the brain. Spinal nerves, they ramify from the spinal cord. Here we have the brain, which includes the cerebrum, cerebellum, and the medulla oblongata. What's the function of these three parts? Is to analyze received information, integrate them, and then you have to take the proper decision. While function of the cranial nerves and the spinal nerves, cranial nerve, they ramify or they branch from the brain. Spinal nerve, they ramify from the spinal cord. They carry messages between the CNS, central nervous system, and the effector organ or the receptor organs. So nervous system is composed of two parts. One, central nervous system. Two, the peripheral nervous system. Central nervous system composed of brain and spinal cord. Brain is composed of three parts, cerebrum, cerebellum, and the medulla oblongata. Why? The peripheral nervous system composed of cranial nerves and the spinal nerves. Cranial nerve, they ramify from the brain. Spinal nerve, from its name, they ramify from the spinal cord. They carry message between the central nervous system and the effector organs. Here we have the nervous system is a network that includes, what does it includes? Includes stimulus. First, we have the receptor organ. Second, we have CNS, central nervous system. And finally, we have the effector organ and to reach the action. So first of all, we need the stimulus. For example, heat. This is a stimulus. Receptor organ, I hold a hot cup of tea. The stimulus is the hot cup. The receptor organ is my hand. The receptor organ sends the message to the central nervous system. The central nervous system sends the message back to the effector organ. It's so fast. The effector organ takes an action to hold my hand, for example, or remove my hand from the cup. First, our receptor organ, it recognizes the stimulus and stimulate a message. My hand, it recognizes that this cup is so hot, you have to do something for this. Second, we have a sensory pathway between the receptor and the central nervous system. It's called the sensory neuron, or it has another name, is afferent. It conducts a message from the sensory organ, which is the receptor organ, toward the central nervous system. Second, we have the central nervous system. It receives information, analyze them, and integrate them, and decide the proper action, and send this action toward the effector organ using a motor neuron. So in the middle here we have sensory neuron, And here in the middle we have something called motor neuron. So motor neuron, it conducts a message from the central nervous system to induce a response toward the effector organ, which has to induce the response or the action. This is by the by the help of the motor neuron, which is called efferent. So sensory neuron between receptor and the central nervous system. Between the central nervous system and the effector, we have a motor neuron, which is called efferent neuron. And finally, the effector organ, it receives the decision to perform an action or to stop an action. So this is the figure. Receptor organ, they recognize the stimulus and simulate a message. Between the receptor organ and the central nervous system, we have a sensory neuron. It's called afferent. Conduct a message from the receptor organ or the sensory organ toward the central nervous system. Central nervous system receive information, analyze, and integrate them toward the motor neuron by the help of a motor neuron. Motor neuron is efferent, which conduct a message between the central nervous system and the effector organ. So between receptor and central nervous system, it's a sensory neuron. It's called afferent. While between the central nervous system and the effector organ, we have motor neuron, which is called efferent, to have the action finally. This is the figure of the spinal cord. Here we have, for example, I may give you this figure in the exam. You have to label it white matter, and this one is called the gray matter. We have the motor neuron, monosynaptic synapse. We're going to talk about this later. So this figure shows the spinal cord. We have the white matter, as I said. Here we have the gray matter. Here we have the dorsal fissure, dorsal root, the ventral root, and here we have the ventral fissure. And then in the middle we have the cerebrospinal canal. Here, as I said, here we have the sensory input, send the message to the brain, integrate, analysis, and through the motor neuron, which is called efferent, toward the effector organ to take an action. So we have, I saw a cup of water, I saw it using my eyes, through the sensory neuron, toward the central nervous system, they are called afferent, toward the central nervous system, and using the motor neuron, which are efferent neuron, toward the muscles of the hand to take an action where the hand approach. So stimulus, receptor organ, sensory neuron, central nervous system, meaning the interneuron, motor neuron, effector organ, and finally the response or the action. So this is the pathway of the nervous message. It moves stimulus, receptor organ, sensory neuron, central nervous system, motor neuron, effector organ, and finally to reach to the response or to the action. R-sim reflex arc pathway. First we have R, which is receptor neuron, they receive the stimulus. Second, S, first R. Here we have S, which is sensory neuron, they pass the impulse on. I, it's called the interneuron between the sensory and the motor, as the spinal cord processes. And finally we have the M, which is the motor neuron which have to act. It is the R-sim reflex. Anatomy of a neuron. A neuron, as we know, is typically made up of dendrites, cell body, which is called the soma, which contains the nucleus, and we have the axon, which may or may not be covered by myelin chain. Finally, we have the terminal arborization, which is called the terminal buds or knobs. So dendrites, cell body, axon. Cell body is called the soma, which contains the nucleus, dendrites, cell body, axon, and finally we have the terminal arborization here. So this is dendrites. Here we have cell body, which contains the nucleus. This is cell body, it's called the soma. Some neurons, they are myelinated, they contain myelin. This is the axon. And finally we have the synaptic terminal or terminal arborization. So the neuron is the basic structure unit of the nervous system. Dendrites, they carry impulses toward the cell body. They carry the nervous message toward the cell body. And axon, they carries impulse away from the cell. We have myelin chain. We have synaptic terminals. They contain mainly epinephrine, norepinephrine, acetylcholine, or GABA. We're going to talk about this later. So The structure and the functional unit of the nervous system is the nerve cell, or it's called the neuron. A neuron is referred to be as a bundle of nerve fibers. A nerve is a bundle of neurons. So a group of nerve fibers, they give a neuron. Group of neurons, it gives a nerve. Group of nerves, they give the nerve system. Nerve fibers, group of them, they give a neuron. Group of neurons, they give a nerve. And group of nerves, they give a whole nervous system. Neurons can be classified following two criteria, either their structure or their function. Classification according to their structure is classified into three kinds. First one is called a unipolar. It's called a T-shape. It looks like a T-shaped neuron. This type of neuron is found in the sensory neurons of the spinal cord, containing one axon, one dendrite, and they are directly joined. Second, we have the bipolar. Bipolar is found mainly in the retina of the eye and the cerebellum, contain one axon and one dendrite. They are joined through the soma. It's called bipolar. And finally, we have the multipolar. Multipolar neuron is mainly found in the cerebral cortex, retina, and the spinal cord. It shows many dendrites, as shown here. It shows many dendrites and only one axon which leaves the cell body. According to their function, we have three types. As I said before, we have sensory neurons called the afferent, receives and conducts sensory information from the peripheral sensory organ toward the CNS, which is the central nervous system. Associated neuron is called the interneuron. It receives nervous message from the sensory neuron, integrated, and then carry or conducts nervous message toward the motor neurons. Look like a link between the sensory and the motor. Mainly it's found in the brain or the spinal cord only. Motor neuron is an efferent neuron. It carries nervous messages away from the central nervous system toward affected organ like a gland, a muscle, to induce a response there. This is a figure that shows a brain in the skin, how does it use an action and a response. Here we have the stimulation here at this level. We have stimulation. It moves through the sensory neuron, serenity neuron, toward the brain, and then the brain sends the message back here, the brain sends the message back to efferent neuron toward the organ to take an action. What is the role of myelin? Myelin chain is a lipoprotein. It's the definition of myelin chain. It acts as a protective layer of the axon and as an insulator. Myelin chain is not continuous along the axon. The gaps are known by nodes of Flannery. Actions, maybe, they are classified into two types. We have something known by voluntary and involuntary. Voluntary actions are actions that require thinking, actions that require the intervention of the brain in addition to the spinal cord. While involuntary actions, they are reflexes, they do not require thinking, actions do not require the intervention of the brain. Is a neuron always active? This is the big question. Are we always listening? Do you think that you always listen? Are we always walking? Are we always in pain? No. Sure no. So, what is the big answer? Do you always think? Mostly you don't think. Mostly, sometimes you have to stop thinking. So the neuron or nerve fiber is said to be at rest in the absence of a stimulation. If there is no stimulation, so there is no action of a neuron. Aim of the following experiments is to study the electrical state of a neuron at rest. Here we have recording of action potential. Recordings are obtained on a giant axon of a neuron by using microelectrodes connected to a galvanometer or an oscilloscope mainly. Here we have experiment number 1. We have nerve fiber, axon, and here we have physiological solution. And here we have the oscilloscope. The needle of the voltmeter, didn't deviate from 0. What does this imply? It remains at 0. This indicates that there is no potential difference between across the surface of the axon, or there is no potential at all. at the surface of the action. Whenever it negates 0, so there is no action potential. Moving to experiment number 2. The needle of the voltmeter didn't deviate from 0. What does this indicate? There is no potential in the internal medium of the axis. Both of the electrodes, they are connected to the internal, so there is no difference, so no potential difference. Moving to experiment 3. Experiment 3. One of the electrodes is outside, other is in the internal part of the axle. So the needle of the voltmeter deviated outward from 0 and the graph indicates minus 70 millivolt or delta V. What can you deduce? What may be deduced from these experiments is as follows. In neuron at rest, there exists a potential difference between the interior and the exterior of the nerve fiber. Between the interior and exterior, not between the exterior only and not between the interior only. Between the interior and the exterior of the nerve fiber. Between the interior and the exterior of the nerve fiber. So delta V is less than 0, which is minus 70 millivolts. So the interior of the nerve fiber is negatively charged with respect to the exterior. The resting potential is the potential difference that exists between the intercellular and the extracellular, between the interior and the exterior of media of a nerve fiber that is at rest. So the resting potential is the potential difference that exists between the intracellular inside and the extracellular outside media of a nerve that is at rest. This is the figure, interior, and this is the exterior. This is the plasma membrane, and this is microelectrodes inside the cell. And here the microelectrode is outside the axis. Okay, the resting potential. What is this meaning? What is the membrane potential? And what the meaning of resting potential? There is an electric charge across the membrane. This is the membrane potential. Whenever you have electric charge across the membrane, that's called membrane potential. The resting potential, when the cell is not firing, is at rest, is 70 millivolt. Delta V is equal to minus 70 millivolt between the inside and the outside. So the resting potential of a neuron, you have to know this, it's minus 70 millivolt. Resting potential, all cells, not just excitable cells, they have resting potential and related to charge across the plasma membrane. The size varies between the cell, but it runs about minus 70 millivolt. Here, you have to know the note that the resting potential may range between minus 5, to 90, minus 90, depending on the species and the nature of the nerve. Here we have 3 Na plus, you are picked up, 3 Na plus are picked, and they pump to the extracellular fluid, and 2K plus you are picked up. So the 3 goes out of the cell and 2K plus they enter the cell. Then the 2K plus are pumped inside the cytosol. How is resting potential maintained? At rest, sodium gates are closed. Na plus gates they are closed. The brain is 50 times more permeable to K plus potassium ions, causing them to leak out. This causes the outside of the brain to have an abundance of positive charge compared to the inside. The inside of the membrane is negative compared to the outside. Sodium potassium pump, Na plus K plus pump, allows K plus ions in for Na plus to go out where 3 Na plus goes out. This further creates a charge difference. But is a neuron always at rest? Some notes you have to keep in mind here. What the meaning of an excitable neuron? A neuron that's able to respond to an effective stimulus is called an excitable neuron. What the meaning of an action potential is term given to the electric visualization of the response of a neuron. It's called action potential. First, excitable neuron, and second, we have definition of action potential. You have to keep in mind also what is the meaning of threshold of excitation. Threshold of excitation if the minimum intensity of a stimulus is able to stimulate a receptor that may or may not emit a nervous impulse or message. Below this intensity of stimulus, no response will be recorded. Now we have to know that upon excitation, a change in the potential difference across the interior and the exterior of the nerve fiber should occur and reach a threshold of depolarization in order to initiate an action potential, which means a response. Threshold of depolarization is the minimum change in the potential difference that should be reached in a nerve fiber so as to create an action potential. Also, you have to keep in mind these notes: minimum time of excitation, the minimum time during which an effective stimulus should be applied in order to excite ganglion. This is some experiments are done. At rest, it's minus 70 millivolt. This is the resting potential. No change in the potential difference upon stimulation or stimulus of intensity I1, which is less than the threshold of excitation. So no change in the potential difference between the where I1 is less than the threshold of excitation, no change in the potential difference, as if nothing is occurred, no action potential is stimulating this nerve fiber. This stimulus is said to be ineffective since it doesn't cause any action potential. Moving upon application of stimulus I2, less than threshold, but you have to say that I2 is greater than I1. You have small artifact here, small action potential, not considered as action potential, artifact. So this is reaching the threshold of depolarization, doesn't reach this. So slight change in the potential difference, slight depolarization, doesn't reach the depolarization. Slight depolarization is called artifact that did not reach the threshold of depolarization. Then everything goes back to the normal situation. This stimulus is said to be ineffective. 3. Upon application of stimulus of intensity I3. I3 is greater than or equal to the threshold of excitation. As sure, I3 is greater than I2, so we have small artifact and we have an action potential here. This is the action potential that I'm talking about. So I3 is greater than or equal to threshold of excitation, and enough change in the potential difference occurs that reaches the threshold of depolarization. An action potential of amplitude about 120 millivolt was recorded. This stimulation is said to be effective, and it's called the threshold of excitation. While I4 is greater than or equal to the threshold of excitation, also an action potential is recorded. As enough change in the potential difference occurs that reaches the threshold of depolarization, the same response was recorded, so the stimulus is also effective. Here we have action potential in nerve fiber. We have different stages: 1, 2, 3, as named here, 4, 5, 6, these stages, and finally 7. What do we call each stage? 1, it's called the resting potential. 1 is called the resting. High popularization or artifact, whenever we have a small high popularization, it's called the artifact. Depolarization, whenever it reaches toward the action. Inversion of polarity, whenever it goes back like this. Repolarization, hyperpolarization, and finally we have to go to the resting potential. This is stage number 7. Here we have some phases of the action potential. 1. Electric interpretation of the difference. Depolarization. What happens? It corresponds to the hypopolarization of 120 millivolt. We have 50 minus 70. It's 50 plus 70, so it's 120. This indicates that the interior becomes more positively compared with the exterior, which should become more negative. Repolarization, when it comes back to minus, which corresponds to the return back to the initial stage. This potential difference drops from 50 millivolt to reach -70. This indicates that the nerve fiber is once again more negatively on the interior with respect to the exterior, which become more positively charged. 6. Hypopolarization. Hyperpolarization corresponds to the increase in the electronegativity of the interior of the nerve fiber, where delta V drops to about -70 millivolt. This indicates that the interior is now more negative compared to the resting state. Now we're going to move to the understanding the ionic changes. Resting phase. This state the cell is highly membrane to K plus than to Na plus, where more K plus on the inside and more Na plus on the outside. Once continuing with the ionic interpretation, depolarization, the membrane permeability to Na plus is more permeable to Na plus where it increases, and the permeability to K plus decreases. It's become more electropositive. This before depolarization, more positively charged on the outside, more negatively in the inside. This figure rapid depolarization where more positive enters inside the cell and the delta V will increase. This is the action potential and the nerve the threshold potential resting about -70, and whenever we have this speed it's called an action potential. So threshold potential will trigger an action potential or a nerve impulse. We're gonna have an action potential or a nerve impulse. So the action potential is an all or none. So read the law of all or none. Moving back to the ionic interpretation of the repolarization, the anterior where it becomes more electronegative. Repolarization phase, the sodium ion channels they close and become refractory. Depolarization trigger opening of voltage gated potassium ion channel. This K plus flush out of the cell, repolarizing and then hyperpolarizing the membrane. This is the repolarization when it comes back toward the negativity. Finally, we have the hyperpolarization. K plus channels are still opening. They are too slow to close. So more positive accumulate on the outside as of the fiber as compared to the resting. The interior becomes more electronegative than the resting phase. Resuming the resting potential, potassium channels they close. Repolarization reset sodium ion channels. Ions diffuse away from the area, and the sodium potassium Na plus K plus transporters maintain polarization. The membrane is now ready to fire again. All or none response. If an action is stimulated above its threshold, it will trigger an impulse down its length. The strength of the response is not dependent upon the stimulus. An action cannot send a mild or strong response. Either it sends or it doesn't. Either it responds or does not respond. Why does the nervous message always propagate in unidirectional? When a zone of nerve fiber is excited efficiently, depolarization occurs and an action potential is created. This depolarization, it will create or stimulate the depolarization of the neurogenic zone ahead. This is because the previous zone is said to be in a refractory period, non-excitable one. As such, the nervous message propagates forward and never backward in vivo. So therefore, the nervous message is said to propagate in a unidirectional manner from dendrite cell body toward the axon and to the next cell. Why? How to measure the speed of propagation of the nervous message? How to measure the speed of propagation of nervous message? Here we have a stimulating electrode and we have a recording electrode. This is delta D, the distance between these two electrodes, and we have delta T is the time between each response. So V, as you know, is equal to delta D over delta T. super distance over the time as in the physics. Factors that affect the speed of propagation. First, the diameter. So we have D1, D2. As you see here, D1, nerve fiber 1, D2, nerve fiber 2. As we all see that D2 is greater than D1, so the nervous message propagates faster whenever we have a larger diameter. So V1 is greater than or less than, since it has less diameter, so D2 has the highest diameter, the greater diameter has the greater velocity. So diameter of the nerve fiber affects, as the diameter of the nerve fiber increases, the speed of the nervous message increases also, where as the increases diameter, the speed of propagation of the nervous message increases. Second factor, myelination of the nerve fiber. Nerve fiber 1, nerve fiber 2. Nerve fiber 1 is not myelinated, nerve fiber 2 is myelinated. So in the nerve fiber 2, the speed is more than the speed in the nerve fiber 1. What does this mean? Presence of a myelin has a higher effect on the increasing of the speed than the effect of the diameter. What does the myelin do for the propagation of the nervous message? In the absence of a myelin in a non-myelinated nerve fiber, the message propagates like this way. While whenever we have a myelin, the message propagates in a myelinated fiber in a such way it's called a way it's called a saltatory movement. Myelin is an insulator. It doesn't conduct a nervous message, so it doesn't conduct. You have to jump over this. If you see here, it's jumping. 1, 2, 3, 4. The jumping movement, as you see here, the propagation of the message is like this way. This is called a saltatory movement or saltatory manner. The message propagates in a saltatory movement, so it's going to be a faster one. Saltatory conduction. The jumping of an impulse between the nodes of round V, thus dramatically increasing its speed. It only occurs in the case of the presence of myelin, 2 meters per second to reach 120 meters per second. Factors that affect the speed, also we have temperature. T1 equal 20, T2 equal 30 degrees Celsius, nerve fiber 1, nerve fiber 2. T1 is less than T2. That means that T2 is going to be the strongest one and will have the strongest velocity. V2 is greater than V1. So as the temperature of a nerve fiber increases, the speed of propagation of the nervous message will increase. Third factor is drugs. Drugs, we have two types. We have drugs that are stimulants. We have other they are tranquilizers. Stimulants, they stimulate the message. Why? The tranquilizers decelerate the